Actually, I just want to add a point here. Our journey in sequencing started in his office room, where before our batch has come, he was dreaming of having a sequencing machine here, and they have given for the order and all. And suddenly we have one day sir just say us ki we have some samples. I promised the government that we are going to sequence it. So who's going to go? And me and my friends, two of my friends, we jumped there, so we'll go. We don't know anything, we just know the word sequencing ka spelling, nothing else. And we were just new into the department. We were just learning RT-PCR from our seniors and our lecturers. We went, we don't know anything. We came back, and that's how Kappa came out. He did it. He just told sequencing in 1 hour, but there's much depth into it to understand. That's the greatness of sir. He makes everything so simple that you just feel, अच्छा इतना ही है क्या? उससे ज़्यादा कुछ भी नहीं है. So that's how it is. And thanks for sir again for that wonderful speech that he has given. I request you all for your lunch, and the next session will be post lunch. You have a tour to the sequencing also, to the sequencing lab of ours during the lunch. So if anyone is interested, you can just let us know. We will take you there too. Thank you.
Where is the lunch?
Lab किसी को देखना है हमारा, तो you सबको okay. So okay, we can take you batch wise there. In the meantime, you can have your lunch too.
Where it is?
Whoever wants to go to the lab first, you can just let us know.
Lunch where it is?
Lunch downstairs.
Yeah,
Downstairs where you had your breakfast.
Go for a lab visit. After you can move to the lab, and after we have official scientific people here. They have their iron current machine. Half of them can have a look at that technology. They are from the official scientific who are going to explain it. So you can divide ourselves. So we will
Lunch
so 25 people can go for a lab visit, and 25 can stay back and have a look at the iron current machine.
Rashmita.
After a heavy lunch, hope you all will be very active in the class, because this is the most important session. After the introduction, exactly what happens on the nanopore sequencing. So this will be the session will be taken by Dr. Rashmita, our third year PG. Oops, sorry, I did that deliberately because yes, she's not a lecturer. She's more like a student, and with us especially. So that's what we feel. She's from JJ Medical College. She has done her post-graduation from there, and her UG is from Bhubaneswar. She is smart, sensitive, sweet, also many S to her. Yes, and she a very subtle lady, but very intelligent, very hardworking. So we look forward how her session would be, and I'm sure it would be pretty great, and you'll enjoy the session. Thank you. Yes, I forgot that, sir. She has won the PM Khare Award, and she 4 times continuously she won the title winner for the PG quiz in Pathologist and Microbiologist Conference of Mumbai. Thank you for that point.
Thank you, Sushma. Thank you, sir. So all participants on board, we have here. So everybody is here. A very good afternoon to all. So already Karyakarte sir has told us or covered the entire sequencing per se. So what I am going to discuss is a journey of a sample right from swab to the sequencer. So what a sample has to go through, or what all stages it passes through, is what we are going to see next. So I divide the entire journey of a sample. First, you have to collect the appropriate sample. Next, you'll have to extract the RNA or the nuclear material what is present in the sample. Next, pertaining to SARS-CoV-2, if I say it's a RNA virus, so what I have to do is to first convert RNA to cDNA, amplify the cDNA, prepare the library, and then load it into a sequencer. So that is briefly what we are going to do in the following few minutes. So the success of diagnosis of any disease depends largely on the quality of sample that is collected, the way it is transported to the laboratory, and the way it is stored in a laboratory. So that is, now what we are going to discuss is sample collection. So what is the sample that I am going to collect? For SARS-CoV-2, as we have seen that there are different number of samples that are shown in this table. However, it is important to note that the level of viral RNA present in each sample is different. So we need to be very cautious when you are selecting the appropriate type of sample. So for SARS-CoV-2 per se, the respiratory sample, as we all know, we all have worked for the last 2 years, we all have been working in a molecular laboratory setup. Everybody has done RT-PCR and everybody knows what is the best sample that we collect for SARS-CoV-2 diagnosis. So it is always the respiratory sample which has got the highest viral load. Next, when should I collect the sample? Whenever the patient presents to you with his symptoms, that is the point in time when you are going to collect that sample. Next, what is the medium of collection, how you are going to store it, and what is the shipment procedure. So in case of SARS-CoV-2, we are going to collect nasopharyngeal sample. We are going to collect it with a Dacron swab. At 2 to 8 degree, it has to be transported to the laboratory, and whenever you are going to ship it to the laboratory within 5 days, it has to be 2 to 8 degree. But if it is more than 5 days, it is always recommended to use dry ice or at -70 degrees centigrade. So what is the medium that we store? That is the viral transport medium. So make sure that you use ICMR approved viral transport mediums, because they are based on CDC and WHO guidelines, and they are free of any kind of inhibitors that can affect the stability of the RNA. Next, so I had to cover the specimen collection. I had to rush through it because we all have been working in a molecular laboratory setup. We know what is the kind of sample that we receive. So next in the step is how we are going to retrieve our RNA. So till today, the most common methods of SARS-CoV-2 RNA extraction is silica-based, magnetic-based, and phenolchloroform. Of these, the first two methods, silica-based method, in very early times of our pandemic, we all have used the silica-based method, but we know, though the quality of RNA is high, it has got a higher quality of RNA, but the throughput of the method is very low. Rather, it's more of a manual kind of extraction system. Then came the magnetic bead method, which gave us a higher quality of RNA sample, but the process was automated. So things were hands-free. We could get more time to, within a very less time, we could extract our RNA. So that is how automation has helped us. Now, once you have extracted your RNA, what are the few points that you are going to look for? You'll have to assess your nuclear material for its purity, for its quantity, and for its fragment size. Now when we speak of purity, that means we suspect there is something that is impure. So what are these impurities? So at the time of extraction, there are various chemical components that are carried over and have got deleterious effects on the viral RNA. We have all gone through the video that was playing that in the early morning. The person was discussing with you. So what we saw was library preparation. So library preparation is a series of enzymatic reactions that take place. You pipette various enzymes, you pipette RNA. So it's a series of enzymatic reactions that takes place. There is reverse transcriptase reaction, there is ligation reaction. So there are various enzymes involved in these processes. So if these contaminants are carried over, they are going to affect or they are going to prevent these enzymes from binding to their target. They are either going to reduce the enzymatic action or they are going to degrade the enzyme itself. So this is how a contaminant in a sample can affect your RNA. So be particular when you are doing extraction. Look for these contaminants. You do not carry over. So this is a huge list of various contaminants, but the common laboratory contaminants that we come across are alcohols. At the time of extraction, we have alcohol washes. So alcohols is one of the very important contaminant in the laboratory. What it does, it denatures the enzymes. So enzymes are proteins, basically we know. So it causes denaturation, thus affecting the enzymatic action. Next are the EDTA, because at many times the illusion buffers are nothing but stress EDTA buffers. So if that EDTA is excess in amount, that is going to chelate the metal ions that are present. Next are detergents during the extraction process. So it also has the methods how you are going to minimize these contaminants. So that is also important. When you have identified your contaminant, you also have to minimize it. So in case when you see there is alcohol present, what you are going to do is you have to dry your pellet. Evaporate all the alcohol that is going to interfere with your reaction. Similarly with EDTA, reduce the concentration of EDTA and then, or you can change the illusion buffer that you are going to use. Next, in case of detergents, wash it with 70% alcohol. Now, once we have checked, once we have identified our contaminant, how we are going to detect these contaminants or how we are going to check the purity of the sample, basically. So based on the principles of turbidity determination or optical density, what we commonly use is a UV spectrophotometer. So what we do is we calculate the ratio of absorption at 3 different wavelengths of UV light, that's 260, 280 and 220. So for a pure RNA, your 260 by 280 nanometer ratio should be approximately 2.0. And for a pure DNA, it should be approximately 1.8. So 260 by 230 nanometer, it should be 2 to 2.8. So why this is important? Because if your sample is impure, if your sample contains any contaminant, it is going to cause overestimation of your nuclear material at the time of quantification. And also, as I said earlier, these contaminants are also going to deter or negatively impact your reaction. So next, now that we have checked the purity of the sample with a spectrophotometer, we have ensured that our RNA is pure. It is 2.0 approximately, has an absorption value of 2.0. Next, what we are going to do is we have to quantify the RNA. So after extraction, what is the amount of RNA that we have eluted, or how much material do I have with me to carry it forward? So for good sequencing yield, and every protocol has got a minimum recommended input volume. So for example, if you are dealing with larger genomes, it is minimum, they recommend you to minimum carry 100 to 500 nanograms of DNA or RNA. And if you are dealing with smaller genomes, it is mostly around as low as 1 microgram. So how are you going to determine, so what is the quantity of RNA after extraction I am going to take forward? So this is, usually what we use are fluorometric dyes or quantitative PCR. So fluorometric dyes, based on one of the example is cubit assay. So what happens is your fluorometric dye goes and attaches to the double-stranded DNA that you have. It intercalates between the strands, and this fluorescence, when it attaches to the DNA, the fluorescence that is emitted out of the sample is what is quantified, and that gives you a quantification of how much nuclear material that you are going to take forward. So incorrect quantification would mean that we are proceeding with less quantity of material than intended, which will lead to poor performance and less output. The third point, how are you going to assess the fragment size? So what is the size of your nuclear material that you are going to carry forward? So the most common methods, gel electrophoresis, we all know, we all have been working in molecular laboratory. And the other thing is a use of a bioanalyzer or a tape station. So this particular machine is nothing but a miniaturized electrophoretic machine, which is a chip-based electrophoretic machine. So what we do by using a bioanalyzer, it is used to assess whether your RNA or DNA is degraded. By degraded, I mean it is reduced to smaller sizes. So the more it is degraded, the poor is the quality of your sample. Another thing that a bioanalyzer tells us is something known as RNA integrity number. So what is this RNA integrity number? It is nothing but a measure of level of degradation of RNA in your extracted sample. So this value ranges from 1 to 10. So 10 being the least degraded sample. So lower is your, the recommended RIN or RNA integrity number, it should be always greater than 7. So as you can see in these graphs, as the RIN decreases, the number of aligned reads, the read length and the yield of the sample decreases. So these are the terms that you are going to come across as we go further and we discuss the library preparation. So the point that you have to keep in mind is that you have a RIN of greater than 7. And what is the instrument that is used to calculate this RIN or assess the integrity of your sample is a bioanalyzer or tape station, or if that is not available, it can be easily done on a gel electrophoresis. So next, once I have obtained my RNA, how do I store my RNA? So the two things that is important is temperature and the buffer that you are using to store it. So the common practice is to store at -20. If you are storing it for a longer period of time, for months and years, it is always recommended to store it at -80 degrees centigrade. The buffers, which are the most commonly used buffer, is nucleus-free water, precedillier buffer, or commercially available RNA storage buffers. So these are the graphs which show you at different temperature when we store this RNA at different temperatures. That is, one at room temperature, one is 4 degree, and one at -20. And we have compared all the three buffers. So as you can see, when it is stored at room temperature in water, the stability or the RIN value decreases. This means when you store your RNA at room temperature in water, you are going to degrade your RNA. So the quality of your RNA is going to decrease. So it is always advisable to store your RNA at -20 degree and above. For longer storage, it is always recommended to store at -80 degree. Now next is, how do I transport my RNA? So we all have been a part, at some or other time, we have been a part of the surveillance SARS-CoV-2 surveillance activity that is going on. So we all must have sent samples to our nearest sequencing center. So how do I send my sample, or how do I transport my sample? So whenever you are transporting your VTM as such, make sure you do it at 2 to 8 degrees centigrade. That is, you can use your ice packs, triple package your sample, use your ice packs and send it. But when you are doing it, when you are transporting your RNA, make sure you always do it at -70 degrees centigrade or using dry ice. Okay, so that is the recommended procedure. Maintain cold chain. Label your samples properly, because that is how you are going to if you wrongly label your samples, we know one sample, we are going to take that sample for sequencing, that the data will not match. So you get wrong results. So another thing that I would like to emphasize is metadata. So whenever we are sending a sample to a sequencing center, one of the most important aspect is a metadata. So what is a metadata? It is nothing but any additional information that can be collected at the time of collection of the sample that has to be sequenced. So in our routine lives, we deal with clinical samples. So what kind of metadata are we expected to collect? It is nothing but your patient age, gender, where does he reside, what was the time of sample collection. So what was the type of sample that was collected. So these are the information that is required. So why are these information important? Because they help us to establish the link between the sequence and the sample that is collected. That helps us to understand the epidemiological pattern of a disease. If a sample that I am sequencing is associated with any outbreak or any cluster or belong to a same cluster. So that is how we are going to relate our sequencing data with the sample. So this is a WHO recommended metadata standard at the time of sample collection. So there are 3 tiers, tier 1, tier 2, tier 3. Those which are highlighted in red are a must and they should always accompany a sample. So what does what does it contain? What is the type of the sample that I am collecting? Any unique identification number, as in case in India we have our ICMR IDs, where it is linked, where each sample is linked with an ICMR ID. Once we have that, we can retrieve the entire information. What is the sample collection date, age, gender, address of that person, the country from where it is collected, originating lab, sequencing lab, sampling method, and the host. So these are the information that is must that we have to collect during sending a sample to the sequencing lab. Another important thing that I would like to highlight about metadata is WHO has laid down criteria to declare a particular variant as a VOC. So what are those criteria? They are based on increased transmissibility. If the patient is if the patient requires any hospitalization, increased oxygenation, or is not responding to any kind of public health measures. So how are we going to decide if we do not have a proper metadata, if we do not have the information relating to this sample, how are we going to declare a sample as a VOC or a VOI or a variant of high concern? So that is why it is what is the importance of a metadata. I am emphasizing it from time to time because in our daily practice we have found it difficult to obtain these patient related information. We used to get the samples from the private labs or any lab sending samples to us for sequencing without any metadata. So when you send any sample without any related information and we are going to sequence it, the information that we get out of it is of no use as we cannot implement it or we cannot analyze it or we do not know what is the importance of the sample. So that is why metadata of a sample is important. Now at the time of SARS-CoV-2 pandemic, what was our sampling strategy? So we had to screen the international travelers. Why? Because we wanted to know if there is any entry of new variant or mutant. We did community surveillance through sentinel labs because we wanted a good representation of various geographical territories to monitor how the virus is evolving in the community. You all must have sent 15 samples every 15 days to a sequencing lab. So that is how community surveillance is being conducted in India by INSACOG. Any special scenarios like vaccine breakthrough infections, super spreader events, cluster, increased hospital admissions, increased need of oxygenation. So these are the criteria, these are the special scenarios when you suspect that there might be something new variant that I am dealing with, which is causing all these. Next is wastewater sampling to know the epidemiology or the spread of the disease in the community. So to summarize our sample collection, a good quality sample with a high viral load is very crucial for retrieving your full genome of high quality. So what is an ideal sample? An ideal sample is the one which is stored at -80 degrees centigrade and is transported to the proper laboratory, ensuring a cold chain. Now that we have seen what are the various samples that we are going to collect, how we are going to store it, how we are going to transport it, next, once it has reached the lab, what are the stages that it goes through? So here, SARS-CoV-2 is an RNA virus. So we are dealing with RNA. So there are few things that we have to keep in mind when we are handling our RNA in our laboratory. So always use wide bore pipette tips because it will reduce your shearing. So the more thinner is the bore, the more pressure it creates and it leads to shearing. Next, gentle mixing by flicking. Don't mix it जोर से पिपेट ऐसे ऊपर नीचे करके वैसे मिक्स नहीं करने का. So you have to, in case when you are dealing with RNA, be gentle with it because we know RNA is labile, it is single stranded, and because of its structure, it is easily degradable. So gently mixing by flicking. So just tap on your Eppendorf tube or where you are storing your sample, just flick it and mix it. Avoid multiple freeze and thaw cycles. Keep the RNA sample on ice as much as possible. It will prevent your nucleolytic degradation and which may affect your sensitivity of the test. Avoid high temperatures and pH that you should maintain. So these are the few good laboratory practices when you are going to do your sequencing experiments. So these are the few things that we are going to keep in mind. Avoid, as I've already discussed, avoid multiple freeze and thaw cycles of the reagents. That time it was of the RNA, now it is reagents. So how do you avoid it? Aliquot your samples. So by doing this, you are not only going to avoid freeze and thaw cycles, but you are also going to prevent contamination of your reagents. So always prepare a 10% volume extra to account to your pipette loss. Clean and UV irradiate your goods so that you have a decontaminated work area. All post-PCR areas should be separate and they should have a dedicated equipment. Aerosol resistant tips to reduce the carryover and contamination. Unidirectional workflow in your laboratory is very important. When you are moving from pre-amplification to post-amplification area, do not move back to pre-amplification area. That is how you are going to introduce contamination. So to monitor your cross-contamination, what you are going to do, what is advisable is always to run a negative control. So that is how you are going to put a check on contamination. And it is always recommended to have a negative and a positive control with every batch of sample that you are processing. So these are the few points that you should always keep in mind whenever you are working in a molecular laboratory, not only for sequencing, be it for RT-PCR test. So the same applies for a sequencing laboratory. Now next, we have collected our sample, we have extracted our RNA. So the next thing that we are going to do is we are going to convert our RNA into cDNA or complementary DNA. Now, the conversion of RNA to cDNA is known as reverse transcription. So can anyone from the audience tell me what all things do you expect for a what all materials do you need for a reverse transcription reaction to take place? Anyone? What enzyme? RT enzyme. So, okay. So what we need here is just an RNA and a Luna script RT supermix. This is a commercial name given to it, but this is nothing but our reverse transcriptase enzyme. It contains an RNase inhibitor which will inhibit the degradation of your RNA, random hexamers, because you have to reverse transcribe your RNA into cDNA. So you need few hexamers, or hexamers are nothing but they are oligodeoxynucleotides of which are 6 in numbers. So you are going to attach it and poly-DT primers. So that is what we require for a reverse transcription reaction. I will not go into the details as in what is the quantity of reverse transcriptase and what is the quantity of the RT mix that you are going to take, what is the input volume. We will only deal with the concepts, why it is done, or what are the enzymes that are used to do it and why it is done. So as you have already seen it in the video played in the morning. What you are going to do is you have to take your RNA in the reverse transcriptase plate or a 96 well plate and put the reverse transcriptase enzyme into it, and then put it set your cycles. So these are the cycles. Everybody is acquainted with RT-PCR, so you know what are the various steps of a PCR. So reverse first will be the primer annealing, cDNA synthesis, reactivation, and hold. So these are the various temperatures that you are going to put, and the output would be your cDNA. So next what we are going to see, now that we have converted our RNA into cDNA, what we are going to do is prepare our library for loading into the sequencer. So what are the various steps? First, what the cDNA that we have formed, we are going to amplify. That is, we are going to increase its number. The cDNA that we prepared from the RNA is equal to the amount of RNA that is present in your original sample. So that may not be sufficient for you to work or to take forward for your at the time of sequencing. So you need to increase its number. So after amplifying your cDNA, we are going to attach the barcodes, we are going to attach the adapters, we are going to clean the library and load it and prepare it for loading into the sequencer. Now how are we going to amplify our cDNA? So what are the materials that would be required? It is nothing but a high fidelity DNA polymerase enzyme, deoxynucleotides, magnesium ions, and the buffer. So this is in particular to Oxford Nanopore Technology Midnight Protocol. Whatever we have also circulated in the circulated manual has the Midnight Protocol. So there are various protocols that Oxford Nanopore has developed for sequencing a SARS-CoV-2 genome. Earlier to this, we were following a Arctic protocol. Now this is a Midnight Protocol. How do they differ? In Arctic, we had a set of 98 primers, whereas in case of Midnight, we have a set of 29 primers. So we have brought down the number of primers. So as a result, what have we done? We have reduced the cost, we have increased the scalability and the speed of the protocol. So what is Arctic? Arctic is a network of people, network of scientists who have been involved in these genome surveillance activities, who have been developing primers for various viruses or pathogens, who have been developing protocols how to sequence them and the pipelines how to analyze the samples. So that is previously we followed the Arctic protocol, which has been now modified to Midnight. This is because the PCR tiling that we are going to do, that produces 1200 amplicons. So 1200, that is the Midnight. So it has been named in such a way, that is Midnight protocol. So what we are going to do along the SARS-CoV-2 genome, as you can see, we are going to put these 29 set of amplicons which are pooled in different tubes, that is pool A and pool B, just to have a uniform coverage over the genome. So these pool A and pool B, they share 20 base pairs with each other. So what is the importance? As you can see, one primer is overlapping on the other. Yeah. Yeah. So these blue ones, these are a set of primers, and these grey ones overlapping on the blue ones. So these are the next set of primers which overlap on pool B. There is a, they share 20 base pair with each other. The importance of these 20 base pair sharing is you'll come to know when at the time of alignment and mapping to the reference genome. So we have the primers, we have the enzymes and the buffers and ions that is required for cDNA synthesis. So I've already discussed, these are a set of 29 amplicons which will produce a 1200 base pair length amplicon size with 20 base pair overlaps. Now next, this is the picture. This is the picture. It shows you have a primer set pool A, primer set pool B. So in case you are processing 24 samples, so you'll have a set of 24 pool A and a set of 24 pool B primers. So there are two set of primers. You are going to set two different reactions. You are going to add the RNA and the primer mix and set the adequate temperature and amplify your cDNA. In the next step, what we are going to do is we are going to attach these rapid barcodes. So what is a barcode? So sir has already told us these are a set of nucleotides which are unique. Every barcode is unique and is produced by and is commercially available. So these are company specific nucleotide sequences which are used as unique identifiers. So how does that help us? It allows a large number of samples to be pooled together. As a result, what is going to happen is we can process large number of samples at a time in a single run. So what is this chemistry actually based on? So these are based on transposes. So the transposes are enzymes which are going to produce a nick at a particular point of DNA, and at the same time it is going to attach these barcodes at these places. So it is going to fragment the DNA and attach the barcode. So this process is known as tagmentation. So this chemistry is based on transposes, enzyme based tagmentation. So this, as you have already seen, what you are going to do is, initially you had primer pool A and primer pool B. You had set two different reactions. Now what you are going to do is you are going to put primer pool A to primer pool B. Now you are going to combine both the pools. We had two different set of primers. Now we'll combine both the primers and prepare a single pool. Now next we are going to attach barcodes to each of the sample. So once you have attached your barcodes, now that your samples are barcoded, we have put all the samples together in a single pool and we have prepared a library. So what we are going to do next is we are going to clean the samples. So how are we going to clean it? We are going to clean it with the use of Ampure XP beads, or these are known as SPRI beads, which are paramagnetic materials which are used to purify your or clean your library. So a point that we have to keep in mind, so, what these SPRI beads contain. So they have these paramagnetic beads along with polyethylene glycol and salts. So these ratio of polyethylene glycol and salt along with what is the ratio of your beads to sample determines what is the base pair or what is the size of the DNA that you are going to discard. So more is the polyethylene glycol in the solution with respect to DNA or RNA. So more it is going to bind to the smaller fragments. So you have generated a 1200 base pair amplicon size, 1200 base pair amplicon at the time of cDNA amplification. Now you are going to clean it. Any kind of impurity, as I said, any kind of impurity is going to negatively impact your library preparation. So at the time of cleanup, you are going to remove these smaller fragments. So the ratio of beads to sample is important. Now once you have cleaned the sample, I think we have already seen this video earlier. So do I need to show it again?
Yes. Yes.
So what you can see here is these are the paramagnetic beads. You have to properly mix them, bring it to room temperature, and then mix it with your sample. So you have to, something that you have to keep in mind is what is the ratio of your bead to sample. So that is going to determine what is the size of the DNA that you are going to discard or clean. So you incubate it in a hula mixer. You mix it properly and incubate. So once that is over, you have incubated for a given amount of time as recommended in the protocol. You put it on a magnet. As you can see, the fluid is clearing off. The magnets are attached to the sides or the walls of the Eppendorf tubes. And the fluid has cleared off. Now this is a very critical step because it is at this step you may lose your DNA that you are going to work with. So you have to be very particular what is the ratio of the bead that you are using, because now the fluid is cleared and we are going to discard this. So you have to prepare 80% of freshly, you have to use 80% of freshly prepared alcohol. You have to wash the magnetic beads with alcohol or ethanol. You have to make sure you wet the beads with the ethanol. It is clearly visible. You can see the magnetic beads that are attached to the walls. alcohol or ethanol, and you make sure there is no residual ethanol in the tube. So because as already discussed, ethanol is a very common contaminant. It is going to denature your proteins. Now that we have completed that incubation period, we'll again wash off the ethanol. So we are trying to remove any residual ethanol that is present, and we'll leave it open to dry so that the any residual ethanol that is there evaporates. Next, with the help of illusion buffer, we are going to add the illusion buffer to the magnetic beads. The size of this RNA that was desired is now attached to the beads. So what we are going to do is now we have to elute the RNA from the beads with the help of illusion buffer. And wait for the fluid to clear off. Now the fluid that is collected is our desired material. So we are going to take forward this RNA. Oh, sorry, DNA. So we have eluted our RNA. What we are going to do is attach the adapters. As sir during his lecture he said, what is the role of an adapter? So we have a small video here. So probably the video is not working. So what is an adapter? So these are also they are small sequences which are attached to the end of the barcoded sample. Along with the adapter, what is attached is a motor protein. So as you can see, the purple color thing is the motor protein. So when a DNA is caught at the flow cell, at the floor of the flow cell, this is the motor protein that goes and attaches to the surface of the nanopore. So this motor protein protein, they contain enzymes which will roll off, or it will remove the double-stranded DNA. It will allow only a single strand to go in, and it will discard the other half. So at a time, a single strand of DNA that goes through the nanopore and the sequence. So we have attached the adapter to the flow cell, and now our library is ready for loading. So these are the basic steps what we do in the sequencing laboratory to prepare a laboratory. So to revise, first we have converted our RNA to cDNA. This cDNA is then amplified. We have attached our barcodes. They are nothing but unique codes that is given attached to a sample just to identify them. Then we attach the adapters. We clean up the library. We perform the library QC. Library QC is done using qubit assay. We have already discussed it earlier. We perform a platform QC. Platform QC is now flow cell check. That would be probably dealt by Justin if he is here. Okay. So yeah, we'll be discussing it in some time, how to perform a platform QC. We'll prime the flow cell and then load our library into the flow cell. So these are the basic steps what we perform to prepare a library and sequence it.
The international speaker, Professor Justin, he's actually traveling from Goa, so by road. So he's stuck up with the traffic out on the highway. So the loading of the flow cell will be continued by Dr. Rashmita. So once we distribute the flow cells to you, she will take you through the journey. Thank you.
Is just routine pipetting. The only thing is that it is very small. So the control of your hand and the care you take while pipetting is the important thing. The only thing that is very different is the flow cell and how to load the flow cell. Loading of all companies as flow cell have different procedures. This one is important here in Illumina, just to make a hole and put the things in. It's very easy to put it in Illumina sequencer. But here what happens is, you have to understand very closely. You will see that there is an area where you load the sample. And it's a very small area. And it has, as I told you, an impermeable membrane on which you have the nanopores. All these nanopores are proteinaceous in nature. They are all protein, living things. So if you put a bubble on it, air on it, so while pipetting, if some amount of air goes in, that air would cover a particular area on that particular trunk. And then what will happen is, the protein will dry off. Once the protein dries off, the pore is lost. So more bubbles you put in, the more area of nanopore is lost, and your sequence is going to be hampered because the number of pores would then reduce. So the sequence qualities and everything would deteriorate. So the most important thing with nanopore sequencing is how you load the sample, and before that how you put the buffers in. Where you have to see that you don't put a single bubble of air in it. Air goes in, your nanopores go off. So that is the important thing, and therefore this is the critical step in nanopore sequencing, and you need a hands-on workshop on that. Because if you master it, sequencing is then a matter of pipetting. That's all. Right? It's that simple.
How to avoid mobile?
For that, what we do is we pull out any air which is there inside the pipette before you load it, so that you are sure that there is no bubble between the fluid which you are going to push in and there. Because The moment you start pushing it through a bubble, the bubble then moves and goes over that particular flow cells, over that pores. And the pores become dry. They are protein in nature, therefore they lose. If just try to remember that a bubble is not going to let anything in. No liquid can go in a bubble. So it would encapsulate that particular area, and those pores there would be a waste. They would go waste. So that thing she would emphasize it when she's going to tell you, of course. But then I thought that a time filler from my side would be better.
Has received their packets. So you can open your packets. What you are going to find is a flow cell, insulin syringe, which will act as a pipette for you. So we cannot individually provide pipettes to each and every person. So we have modified this insulin syringe. We have attached a tip to it and made sure that it acts as a pipette for you.
We are going to sell this technology to Nanopore.
Yeah, sure.
It is a very innovative thing which has been done. What has been done is, when we, Nanopore does exactly this. I think if Justin would have come, it would have been better. But they do this every year in the annual conference which Nanopore has. It's called London Calling. So it's in London as Oxford is in London. So they do it every year, and they send these kits generally to all the labs, and there they give a tuberculin syringe, and there it's only the loading which takes place. So what we thought was we'll also show you how to remove the bubbles, and so we have attached a tip to it, and we have innovatively sealed it with a MC. And we are going to sell this technology to Canada. But then Justin isn't here, so I think, yeah.
Your flow cells with you. You have opened your packets. So I should first introduce you to the flow cell first. What are the different parts of a flow cell?
You please have a look at the screen for some time so that we can identify each and every component of the process.
Light off. Light.